Welcome to today's lecture on the exciting field of cell biology, specifically focusing on the intricate world of chromatin structure and function. Chromatin plays a fundamental role in the organization and regulation of our genetic material, DNA, within our cells. Understanding the complexities of chromatin is crucial for comprehending various biological processes, such as gene expression, DNA replication, and even aging. In this lecture, we will explore the different aspects of chromatin architecture, function, and its contribution to cellular processes. Let us delve deeper into the world of chromatin. Chromatin is more than just a passive packaging material for DNA, it serves as a dynamic platform for regulating gene activity. There are several key features of chromatin that we will cover in this lecture. These features include chromatin architecture, linker histones, nucleosome positioning, chromatin compaction, chromatin protein complexes, chromatin looping, chromatin insulators, telomeres, chromatin replication, heterochromatin formation, and the relationship between chromatin integrity and aging. Each of these topics contributes to our understanding of how chromatin structure influences gene expression and cellular function. So, let us begin with the first topic, chromatin architecture. Chromatin is organized into distinct structural units, known as nucleosomes. Nucleosomes consist of DNA wrapped around a histone octamer core, forming a bead-like structure. This organization allows efficient packaging of DNA within the limited space of the cell nucleus. The specific arrangement of nucleosomes along the DNA molecule and their interactions play a crucial role in gene regulation. Various factors, such as histone modifications, DNA methylation, and chromatin remodeling complexes, influence the positioning and stability of nucleosomes, ultimately impacting gene expression. Linker histones play a vital role in stabilizing the structure of nucleosomes and higher-order chromatin organization. These histones bind to the DNA between nucleosomes, bridging them together and promoting compaction of chromatin fibers. Linker histones also interact with other chromatin proteins, contributing to the regulation of gene expression and chromatin dynamics. The precise positioning of nucleosomes along the DNA molecule is critical for gene regulation. Nucleosome positioning is influenced by various factors, including DNA sequence preferences, DNA binding proteins, and energy-dependent chromatin remodeling complexes. By controlling nucleosome positioning, cells can regulate access to DNA, allowing or preventing the binding of transcription factors and other regulatory proteins. Chromatin compaction refers to the folding and packaging of chromatin fibers to fit within the limited space of the nucleus. Different levels of compaction can be observed, ranging from loosely packed euchromatin to highly condensed heterochromatin. The degree of compaction influences the accessibility of DNA to various cellular processes, such as transcription, DNA repair, and replication. Chromatin-associated proteins play a crucial role in regulating chromatin structure and function. These proteins include histone modifiers, chromatin remodelers, and transcription factors. Histone modifiers catalyze the addition or removal of specific chemical groups, such as acetyl, methyl, or phosphate groups, on histone proteins. This modification alters the interaction of nucleosomes with DNA and other proteins, influencing gene expression. Chromatin remodelers utilize ATP hydrolysis to shift, slide, or evict nucleosomes, allowing access to DNA for the binding of regulatory proteins. Transcription factors bind to specific DNA sequences, recruiting other proteins and influencing gene expression. Chromatin looping refers to the physical interaction between distant regions of chromatin. These interactions bring distal regulatory elements, such as enhancers and promoters, into close proximity, facilitating the regulation of gene expression. Chromatin looping is mediated by protein complexes, such as CTCF and cohesin, and can occur within the same chromosome or between different chromosomes. Chromatin insulators are DNA elements that act as boundary elements, preventing the spread of chromatin modifications and maintaining the integrity of chromatin domains. Insulators regulate the interactions between distant regulatory elements, ensuring proper gene regulation. Proteins like CTCF are key players in mediating insulator function and chromatin compartmentalization. Telomeres are specialized regions at the ends of chromosomes that protect them from degradation and fusion. These repetitive DNA sequences are associated with specific telomere binding proteins that contribute to chromatin structure and function. Telomere chromatin undergoes dynamic changes during cell division and aging, impacting cellular lifespan and genome stability. During DNA replication, chromatin structure needs to be dismantled and reassembled properly to ensure accurate DNA synthesis. Various factors, including chromatin remodelers and histone chaperones, are involved in the coordination of chromatin replication. Failure to replicate chromatin accurately can lead to genomic instability and diseases. Aging is accompanied by changes in chromatin structure and function. accumulated damage, altered histone modifications, and changes in nucleosome positioning contribute to age-related changes in gene expression. Understanding the impact of chromatin integrity on aging may provide insights into age-related diseases and potential therapeutics. Heterochromatin refers to the condensed and transcriptionally silent regions of chromatin. It plays a crucial role in gene silencing, X chromosome inactivation, and the maintenance of genome stability. The formation and maintenance of heterochromatin involve various factors, including histone modifications, chromatin remodelers, and non-coding RNAs. In conclusion, chromatin structure and function are intricately linked to the regulation of gene expression and various cellular processes. The topics covered in this lecture provided an overview of the key aspects of chromatin, ranging from its architecture and organization to its influence on gene regulation, replication, aging, and maintenance of genome integrity. Understanding the complexities of chromatin will continue to drive research efforts in the field of cell biology, shedding light on fundamental biological processes and potential therapeutic interventions. Thank you for your attention, and I hope you enjoyed this deep dive into the fascinating world of chromatin.